Hi, everybody. Welcome back to that hit podcast, Bimbo Mentality, a podcast where we talk about cool stuff. I'm Bug. That's Emma. How are you, Emma?
I'm great, just thinking about all the exciting things that we have in store on the podcast in 2026. So,
We have so many cool things in store. I have some exciting ideas, by which I mean like a couple of exciting ideas, but we don't want to spoil the fun. well, we have lots of exciting things coming, like more episodes of Bimbo Mentality. Over in the booth doing producer stuff, it's Lilith. How are you, Lilith?
I'm doing good. I can't eat crunchy things or spicy foods right now, which is miserable, but I'm alive.
Yeah, and you have a cool new tattoo.
That is right. I do. This is why I can't really see it on there it is.
You know that great audio recording moment where you show
Yeah.
the audience a visual image?
Yeah, oops.
For the folks at home, Lilith got an inner lip tattoo that says killer on it. It looks really cool.
it does.
There's going to be an op-ed about us. We're going to be in the next NPR op-ed about how, like, podcasts aren't any fun anymore because they're all doing visual gags. Anyway, we're joined this week on the podcast by our guest, Sierra. Sierra is a molecular biologist and bioinformatician with 9 years of research experience stretching across her bachelor's and 2 master's degrees. Her research focuses have included neurophysiology, genetics, sequencing techniques, and more. Welcome to the podcast, Sierra. Welcome to Bimbo Mentality. How are you doing today?
Hello, I am doing great. Had a lovely little holiday season, and I'm excited to chat.
We're excited to have you here. We are excited to see if you can think like a bimbo and teach us all about molecular biology. So let's prime the pump on what we know about molecular biology, Emma. What's your background with this field? I'm sure you're very familiar with it.
I'm not gonna lie, I actually have pretty much no knowledge of it whatsoever. I'm guessing like really tiny things, right?
Perfect. This is
Yeah.
fantastic.
Tiny living things is what I would guess.
Mm-hmm.
Yeah, I was kind of under the impression that like cells were biology and molecules were physics, but so that's sort of how we're coming in. Sierra,
I'll be real. This is like the one Bimbo Mentality topic where I'm going into it and I'm like, no, hold on. Molecules are too small for biology. That's what the fuck?
Lilith, welcome to the bimbo side of the line.
Well, okay, so I'm almost gonna get started on a side ramble here because there's so many like interrelated fields that are kind of like right at the border of chemistry and biology. And so I say like molecular biology, but the full title of like one of my master's degrees is molecular cell and developmental biology. So like it's a bunch of different components. I think like Emma, you mentioned like molecules are too small. It's not the biology of little things. It's not the biology of little things. It's the little parts of big things. Right?
Okay.
So a lot of kind of what like I'm going to be talking about today is kind of the molecules that are in us humans and what are the big molecules that make up our cells, which make up our bodies. And that's so there's a fuzzy zone between where chemistry starts and biology begins, and we're like gonna be right at that margin there.
Little parts of big things, like an elephant's nuts. Sierra, you did very graciously send me two very interesting looking articles about kind of what you do. And I went ahead and I read the abstracts for those, and I've just whizzed just right over my head. I did not understand what we were doing. There was the word like genome, but it had an E in it, and not where I expected to see it, and I just gave up. That's sort of where I went. So instead, I guess I'll sort of start off the conversation with like a handful of words that I vaguely remember from biology class.
Okay.
Mitochondria, photosynthesis,
Incredible.
phylum.
Incredible. Cool.
Am I in the territory?
So I'll probably be mentioning none of that today.
I nailed it.
Absolutely nailed it. Yeah, but like I basically fully prepared to go from like I don't expect to really get to those papers, but like there's so much like interesting like foundational concepts that are like piecewise slowly built up to those papers.
I appreciated having them available because it's always nice to be reminded of why I am one of the hosts on this podcast. You know, it's important to get that material and sort of bring me back down to my level.
These papers are not like my exact area of research. More so they're like I would say like I have enough knowledge to actually read the papers and actually like get them. But the reason that I selected them was because they both deal with kind of situations in which primary sex tissue changes past development. And understanding a lot of biology like that, as I made the decision to go on hormone replacement therapy, like while I was in grad school, a lot of those papers and a lot of those studies like that showed situations where weird stuff like that happens, kind of led me to do an almost side project of my own research, exploring that and exploring papers like this alongside my education. So it's been very fun for me to dive into these kinds of things and kind of see how it also affects me and how it's reflected in me and other trans people, and it's really cool.
That's so cool. You did kind of set us up for our first question there, though, which is kind of okay. So if that's not what your area of expertise is, what is your area of expertise? Tell us a little bit about what you get up to in the field that you are in.
Yeah, so I would say I'm primarily a bioinformatician, and basically what that means is that I use computers to process large volumes of biological data. I'm usually kind of on the like method side of things. So I'm usually like developing sequencing techniques for genes, for gene regulation, which is pretty much what the topic of this is going to be, and like developing the toolkit for how to study how genes turn on and off over time. I've done this in a lot of contexts ranging from like cellular damage and resilience, to cancer research, to also like cellular development, which is largely what these papers are about. So I'm kind of like, providing the tools to study this kind of thing, even if I'm not directly studying it myself.
I guess I wouldn't say like, I think it's so interesting because I think for someone who has no insight into this entire field, I think sometimes I think about like how you mentioned you're providing the foundation or the framework for others to do things. I think in my head, if I had any concept of what molecular biology entailed, it would be like the people studying it. But I think sometimes probably like, I know I do, I don't think about like who builds the frameworks for them to do said study. And so I think that's really interesting that you're doing that. Like I think it's something that probably most people maybe don't think about when they think about like any kind of field, right? Like, but I'm sure there are other folks analogous to what you're doing in a variety of fields as well, kind of in similar fashion.
And on that topic, I'm curious how you kind of found your way to doing that kind of work. What brought you down that into your field?
Yeah. So actually I way back eons ago in the far off time of 2016, I started an undergrad as an ecology major. And like I was more interested in like the environmental science and ecology side of biology. And just like the more I kind of like got into it, the more I realized that like the kinds of questions I had about the world and about biology always like led me to molecular biology. Like, oh, you ask like, oh, how can something survive in a hot spring, you know? And then suddenly you're dealing with all of these molecular and biochemical phenomena to explain something happening out in nature. Oh, how did these like two bird species evolve and diverge from each other? And then suddenly you're thinking about DNA as a molecule and how it's shaped, how species adapt to different environments. And I slowly kind of like got pulled deeper and deeper into that world. And I started spending a lot of time, you know, around molecular biologists, and I eventually changed my major. Um, the bioinformatics part of it basically comes because I knew a tiny bit of code around a bunch of biologists that didn't know code. So they kept on asking me to be the computer person for the lab, and I just slowly developed that toolkit over time. That's kind of how I got here.
This is why I could never be a scientist, because I gotta say that when I think about like why did two different birds look different, my brain does not go to like the DNA chains involved. That is not what I end up thinking about. I just kind of go like, that's life, I guess, and move on to the next thing. So, sort of step one, kudos for following through on curiosity. That is bimbo mentality. Even if people may not think that biology is bimbo mentality, but we're here to prove that it is. Um, and yeah, I mean, kudos for just kind of taking on a role that nobody else was willing to step up and do, because that's a big part of just how society works.
I think, like, I don't know. I think it's like, on the one hand, I want to be like, oh my god, I'm so smart. Look at me. Oh my god. Um, on the other hand, like sometimes I talk to like a real computer scientist and I get humbled really quickly because I'm kind of reminded that like I like biologists are kind of stupid as far as computers are concerned. And so the only reason I've been able to carve out this niche, God, I really hope no biologists are listening, but like the only reason I've been able to like carve out this niche is just because I existed in this environment and I've been perpetually like fixing my grandma's printer or something, except for like people with PhDs, is kind of how I think about it. Like a lot of the actual analysis I do is like I would say pretty simple, but like the application of it to the unique situations is something that like I've been granted the opportunity to do with people who are very intelligent in terms of these biological problems that have taught me a lot, but are gratefully really, really stupid as far as computers are concerned. So I've been able to wedge myself in there.
One of my favorite things that happens on this podcast is when like whole sectors of an industry just get a bunch of shit slung at them. I think this happened on the mechanical engineering episode with Quinny too,
Yeah.
that I tried to start some kind of mechanical engineering civil war, and now I'm gonna try to do the same thing here. So get ready, every biologist.
Hey now, I also hate the computer science side of the field because all the computer scientists are biology stupid and all the biologists are computer science stupid. And I am stupid in both, but still smarter in both than than either of them is in their less specialized field. So
So we're kind of doing a Romeo and Juliet thing, but where instead of being about like teenage love, it's about typing on the computer.
Pretty much. It's about feeding a bunch of cells in it's about a bunch of biologists growing a bunch of cells, getting data on those cells, and then throwing that data at computer scientists and getting them to do things. And yeah.
Okay.
So you kind of have to be
now-
like, you kind of have to have like the like liaise with people and have like the people side and the tech side sort of to like marry those two worlds in a way.
I guess. I mean, I'm definitely like been far more in a biology environment. Like, all of the things I do are like very applied studies to particular biological problems. And so I'm usually talking with biologists, and I usually like am mostly interacting with them, and I feel like I know how to interact with them at this point. But I mean, I guess I am one too, but whatever.
Well,
I love that.
okay, so now after sort of 15 minutes of just kind of causing industry strife, that did lead me to a real question. So let's actually talk about molecular biology. You mentioned collecting data on cells. What does that mean? What does that entail? What does it mean to collect data on cells?
Perfect. I can go into kind of like a really foundational thing, which is, have any of you heard of the quote unquote central dogma of molecular biology?
I am gonna tell you right now that I have not heard of that.
Yeah, no,
Beautiful. So yeah, this is an extremely oversimplified version of kind of this core concept. And it's this core concept of like, how does information that is passed from one generation to the other in the form of DNA, how does that become a functional effect? Like, what are the actual like molecules involved in doing that? And so the reason I bring this up is because there's a very specific stage in this process at which you're kind of capturing something to collect that data on. I hope you've heard of like DNA and the concept of like a gene.
I've watched Jurassic Park, so yes.
Cool.
Yeah, yeah. The DNA is the one that's kind of like a swirly, like a spirally thing.
Bingo, Dino DNA. Yes, I've
You
watched
know what?
that
That's
one.
all we need to do. Podcast over. DNA is swirly. Bye. No. So, DNA is a molecule whose kind of primary purpose is storing information and also helping pass that information along to the next generation. DNA, you can kind of think about it as like a word with letters, where the only available letters are A, T, C, and G. It has a directionality, so you can actually read it kind of in a like left to right fashion. And so a snippet of DNA that is a functional chunk, a piece of DNA that is going to do one thing, is what we call a gene. Genes are really important because the general idea behind the central dogma of molecular biology is one gene, one protein. So what happens to the DNA is that chunk of it, a little like reader molecule comes in, and it kind of reads that chunk of DNA, and it produces a copy of that chunk in a slightly different form called RNA. Now, DNA's purpose is partially to be static, right? If you're familiar with computer like hardware, it's like the hard drive. It's storing all of this like permanent information. RNA is a piece of it that is like floating. It can go wherever, it can do wherever. You can produce it or you cannot produce it, right? So you produce this piece of RNA that contains all the same information as DNA, but it can now do other things. And the main thing that it's going to do is it's going to go over to another piece of molecular machinery, and it's going to get copied again, but this time into something that is very chemically different called a protein. So you probably heard the word protein, but what a protein
that's
actually
in one.
is,
That's what's in like fish and stuff.
yeah, that's like creatine powder, fish, and meat. I mean, that's quite literally true. But more generally, like, proteins are the molecular workhorse of your body. Like, most functional things that happen in your body start with some kind of protein. Like everything in your body that is not a protein was probably produced by a protein facilitating a chemical reaction, or it was sequestered or transported by a protein to a specific location.
A workhorse would probably also be a really good source of protein.
Honestly, yeah. Very meaty, very muscly. Hell yeah.
That was a total tangent. You were talking about science.
Yeah. That is like very, very, very loosely. The central dogma of molecular biology is a segment of DNA called a gene produces a floating piece of that same information called RNA, which then produces a functional molecule that's going to do any number of effects called a protein, right? Now, there are thousands and thousands of asterisks to this. This is like, to call this an oversimplification is like under exaggerating it. But this is kind of like the core framework we're working with here. The reason why I say this as an introduction to like what I do is because each one of those stages, you can isolate those molecules, the sum total of all of those molecules that are in a piece of biological tissue, and you can read them. Using certain like technologies, you can essentially take that gene and you can read it like it is a word. There are devices called like sequencers, processes like sequencing that can give you essentially the sequence of those A's, T's, C's, and G's in your DNA. Your A's, not T's, they're replaced with U's, but your A's, U's, C's, and G's that make up RNA, and also all of the different variety of individual molecule components that make up a protein. And based on that readout, you can start to kind of piece things together and assemble a picture of what biological processes are happening in a cell at a particular moment under particular conditions. So for me, the level I work with the most often is the RNA level, which is one of the most interesting because a lot of times genes are essentially turned on or off by deciding whether or not to produce the RNA associated with that piece of DNA. So like one instance in which I think this is like really important to think about is you have billions or trillions. I don't actually know the exact number, but like you have lots of cells in your body.
Like 7 or 8.
At least 10.
At least
Fucking that's crazy.
At least 10. That's fine. Those at least 10 cells all have the exact same DNA in them. It is a complete copy of all of the DNA that makes you, that encodes your body plan and all your traits and all of that.
Well, hold on, but I feel like it's a little bit of a waste to have that many of something that all does the same thing. I think maybe we should just be one really big cell.
Now you're thinking like an algae. The largest single cell in the world is a foot across, and it's a giant green like globular algae. It pops up in like people's aquariums sometimes. I'm sorry, that's a complete tangent.
Here on Algae Mentality, we're here to see if you can think like an algae enough to
just entirely revert to a single cell.
Algae.
Pretty sure Perfect Cell in Dragon Ball Z was like at least 8 feet tall, but I feel like that was the
damn.
A huge
Earth life has got to step it up. I'm telling you.
Is that Cell's thing in Dragon Ball Z? Is he just one big cell? I haven't watched since I was like 12.
I honestly forgot. I just wanted to make a really dumb joke. I'm sorry. Continue. I'm actually learning so much right now. This is so helpful.
Okay. Fuck, where was I? Right, at least 10 cells of the body. And
Yeah, we were talking about how there's a lot of cells with a lot of the same DNA.
yes. But here's the thing. The DNA is not just encoding like you as an overall body plan. They're encoding like very precise cellular functions. They're encoding very precise things about each individual cell. They're encoding very precise reactions to conditions in your environment, right? So if a neuron, let's say a nerve cell, a cell in your brain, has this exact same plan, the exact same information as, say, a muscle cell, how the hell are they different? And this is kind of like where things start to become interesting. Because it turns out, along with each gene, in addition to the sequence of letters that is encoding for a protein of a specific function, there's also a sequence of letters just before it, and sometimes elsewhere. But there's also a little sequence of letters just before it. And that sequence of letters is kind of a controller. It's called a promoter, or more generally, a regulatory region, because there's different kinds of regulatory regions. And that controller is a place where other molecules bind. And those other molecules, and whether or not they bind at that site right next to the gene, help determine when, how, under what conditions that piece of DNA is turned into RNA and then a protein. So your muscle cells have all of the genes required to potentially make them neurons. They have all of the DNA potentially required to make them neurons. But there's nothing in there, there's no molecule there that's telling it, hey, turn this DNA into RNA so it can be a protein, and that protein will then be a neural protein. That's not happening in a muscle cell. So that gene isn't really turned on. It's not expressed, as we is what we would say.
This is a really dumb question, but when scientists grow tissue from stem cells into other things, are they taking those cells and then telling them what to turn into?
That wasn't a dumb question at all. That was incredibly on topic and interesting.
That's kind of exactly what I was going to talk about next.
Oh my god!
Yeah.
Well,
oh my god! Look at
in
me.
a little bit, but yeah. So yeah, that's actually a perfect segue that handles it. So
for the audience at home, Emma just did a little happy dance. I just wanted to share that with
adorable.
the audience because it was really
Emma,
cute.
you got a good grade in podcasting.
Yay!
So, stem cells. What a wonderful concept. So what is a stem cell? So, when two people love each other very much, sperm and egg meet.
All right, well, we've now reached the inevitable part of the Bimbo Mentality podcast where we talk about fucking. So everybody buckle
we're always
up.
going to talk about fucking. Come on.
It just happens to us here.
They form one single cell, right? That cell.
Hot.
That cell is not a neuron. It's not a muscle. It's not a skin cell. It's not anything. It is what we would call undifferentiated. It has not differentiated into anything in particular yet, right? It's going to divide, and those two cells are then going to be okay, well, it's going to divide quite a bit, and then they're going to differentiate a little bit more, where each cell kind of reduces the possibilities of what it could become. Where it's one cell might be well, I can't really become, oh crap, I need to remember the exact tree of it all, but it's oh, well, I can't become a skin cell anymore, but I could become a muscle cell or a neuron, right? And slowly they'll divide and they'll become more and more and more specific until they reach a fully differentiated state. throughout that entire process, Emma, you're like kind of right in your way of thinking. What's happening is kind of this complex interplay where each one of these cells is making decisions about what genes to turn on and what genes to turn off. And so when scientists are talking about stem cells and turning them into different cell types, what they're doing is they're adding some of those molecules to it, some of those like chemical signals to it that are then able to turn the stem cell into a different particular type of cell. And this is why stem cell research is like such a hot button issue, is because if you want to repair some kind of cellular damage or like address some kind of disease, a stem cell is something that can become anything. And so you can use it as a way to make new tissue that can replace any tissue that you've possibly lost.
Stem cell therapy was used to lengthen the career of professional wrestler Ray Mysterio.
I can't believe they made Ray Mysterio's cells fuck.
I remember reading that.
That's...
Hey, I might be fucking wrong because I may have just read a piece of wrestling news 10 years ago or something. I'm going to look this up. His Wikipedia article is very long.
Ray Mysterio.
Yeah. Let's see. Let's see. Yeah.
I love watching Lilith discover things in real time.
Okay. It literally just says stem cell therapy, cocktail IV of stem cells.
It's kind of a mystery which stem cells. I assume there are different kinds. I don't know that we've covered
there
that
are
yet.
So when I said like, oh yeah, each cell is making decisions where it's like reducing the possibilities of what it could become. Each thing that is not like fully differentiated that can still make like some kind of decision about what it could be is like still a stem cell, right?
Okay.
And there are even stem cells in the...
Once it's like embarked on a career, then it's no longer a stem cell.
Yeah. Once it's like committed, you know, like once
you send that stem cell to college, and then it decides on its major, and
once it's decided whether it's going to join the computer biologist faction or the anti-computer biologist faction.
Yeah, and then there are some cells that are lazy bums that don't commit throughout your life and remain stem cells throughout your life.
You know what? I support those stem cells.
Where do those even like live in my body?
Predominantly in the bone marrow. So you know how red blood cells are like so red blood cells are very weird because they're one of the few cells that don't have any DNA. They're kind of like a partial cell. Yeah. So they have no instructions. And so how they're actually produced is there are stem cells in your bone marrow where their entire job is to divide essentially asymmetrically, divide into two cells, and one of them will stay that same kind of stem cell so it can keep living, and the other will become a red blood cell. Red blood cells themselves don't divide on their own. They don't carry information of their own, and they have a relatively limited lifespan in comparison to a lot of other cells. So thank your bone marrow for being the lazy weed smoking bum that can't decide what to do with its life. I love them for that.
Bimbo Mentality, I would like this to be on the record, supports long-term unemployment, both for stem cells and anything else, and is a pro-welfare fraud podcast.
Hell yeah.
Fuck capitalism.
I've just dropped a link in the chat. I found the clinic that Ray Mysterio said he went to, and I pulled up their website, and I pulled up the page, the science behind our...
Okay, it's MSCs.
Yes. So...
What are they?
Can we...
Achieve your maximum cellular healing potential.
I'm like,
okay, crazy idea.
This is gonna capture me recently.
Can we call them live on the podcast?
Oh my god, this website
Well,
is
I'm trying to
something.
determine like how much bullshit this is.
Yeah, because it may
Um,
be bullshit. A lot of things that wrestlers do to their bodies is bullshit. So,
That's true.
Yeah.
They have a section for anti-aging and biohacking, so
Are you telling me that
Ooh.
people who like slam each other's heads with chairs are not super responsible about what they do to their bodies?
okay, recently WWE promoted the first EMF protected professional wrestling match. They had like little devices that are supposed to protect you from EMFs all set up around the ring. It was the biggest bullshit thing you've ever seen in your fucking life.
Oh
I'm
my
sorry I'm
God.
completely derailing this podcast.
Yeah.
But it was
No.
No, hold on. We need to talk about this.
There's like those people who like pay an enormous amount of money to get property in that one like electromagnetically dead zone around
Yeah.
this telescope in West Virginia. So they're basically marketing to those people, I guess.
I just went on the website and I found a section, and they offer a section that says, at RejuveStem, we offer advanced stem cell therapy to address autism spectrum disorder.
oh, excellent!
God.
Fuck yeah! Oh,
Did
that
it do
rules!
ADHD? I could be healed.
Yeah.
But-
Oh my
Power.
god! Oh my god!
Stem cells.
Backtracking
Okay.
just a little bit to the EMF conversation about
Yeah,
wrestling, 'cause I
sorry,
have an important
sorry.
thing to drop here. But this is going to be a little bit of a revelation for some of our listeners. But in addition to being a podcaster, I'm also an amateur ghost hunter. And EMF means like has a sort of is a bit of a trigger word for me in that regard. I'm just really love the idea that professional wrestlers are now protected against ghosts.
Yeah, exactly. This match was
Alright.
completely
Oh my
unhaunted.
God.
The Undertaker could not attack in the ring, even if he wanted to.
Oh my god!
All right,
Man.
getting us back a little bit
Yes,
in the
please.
general direction of the topic.
Okay. I'm gonna close
Sierra,
this website because I'm like, oh man, this is like, yeah, we gotta tell RFK about this. It's not the fucking
Yeah, right?
all right. Getting us back in the general direction of the topic, getting us back onto the topic of sort of serious science and important conversations in your field, Sierra. My next question for you, Sierra, is: can stem cells cure autism?
Stem cells can't cure autism because there's nothing to be cured. I mean, unironically, but also like,
Unironically, yes. This is typically a joke, but Bimbo Mentality is a pro-autism podcast, and people who are shitty to autistic people can fuck off into the stratosphere.
This is an autism haver podcast, in fact.
I mean,
Yeah.
am I the diversity? I'm ADHD, no autism. Am I the diversity guest?
Wow.
You actually, yes, actually,
Yeah,
I am.
I like sometimes I call myself like an autism grumbler because like half my friends.
Oh my god!
So many of my friends are autistic.
All for the audio or for the listeners at home, all of our jaws dropped in that one. Just across
I'm gonna
the
use
board.
that because that's also me.
Oh my
That's
god!
That's really good, but I do worry about us getting off on a whole tangent about grunglers for the second time in 3 episodes. I don't know if we can afford that.
Oh my fucking god! I called a guy I matched on a dating app with a grungler at one point because he mentioned having a lot of trans women friends, and I'm like, oh my god, I found myself a grungler. Anyways,
All right, we're back in grungler territory. We have to talk about
Oh
this.
no.
Okay, wait! I have to know how he responded.
he like was really confused by it. And I like had to send him the original like grungler like meme text, and he's like, "Wait, I am in a Discord server with a bunch of trans women."
Yep.
Gruggler identified.
It's like, I am a bisexual man in a Discord server with a bunch of trans women.
Yeah.
Oh my god.
This is.
Anyways.
I want to be clear about this one too. Bimbo Mentality is also a pro Gruggler podcast. Guys out there who just hang out with us and chill with us, we love you. You're cool.
Bisexual men who fuck with the dolls, I love you.
All right, let's talk
my
about
God.
science.
What the hell was it on? Stem cells, cell differentiation,
Yes.
blah, 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 blah. Okay.
We were talking about stem cells, and then we were talking about ghosts, and then we were talking about Grugglers. You were talking about the
Right,
science of
okay.
stem cells.
So there is okay. We got the idea established that you have a stem cell, and then the stem cell splits, and those become more specific kinds of stem cells, and those split and become more specific kinds of stem cells until they reach a fully differentiated state. Now, most of the time, if you think about that as like a tree, you're only going one direction, right? You're only going towards the more differentiated state. A lot of stem cell research focuses on reverting that. So taking a fully differentiated cell and smacking it back a couple steps so you can have some material to work with, right? Excuse me. That's very difficult to do, and it usually doesn't happen. There's another kind of exception to this, which I've studied this process in other contexts that are not these papers, but we are slowly coming up on the papers. But sometimes, if you think about the end differentiated states, one fully differentiated cell type, under extreme circumstances, without reverting to a less differentiated cell type, without doing anything that's like stem cell-y, if you do something just right, it will just become a different cell type. Another fully differentiated cell type. It is very rare. It is a process known as transdifferentiation. And we know all about them trans. But it's a very rare and very poorly understood process of which there are very few examples. And actually, both the papers I provided are examples of that happening. Because if we think about the cellular development tree, that also includes things like gonadal tissue and primary sex tissue, right? So all quote-unquote female and all quote-unquote male reproductive tissue are essentially differently differentiated states of the same progenitor cells. So there's the same branch of the tree. There's like these undifferentiated gonadal cells that go through a couple stages of cellular development, and then at one point, one of them decides to become testes, they decide whether to become testes or ovaries, right? And then from there, a lot of other development proceeds. Now, so, oh my god, this has got like so derailed that I'm like trying to think about like what I've established.
Now I'm curious to see what you're gonna say, because I'm like gonna be like, if I was born like 50 years from now, could I have like turned my balls into ovaries without having to like, you know?
So okay, yeah. So essentially, yeah, that's kind of what I'm building up to, and that's actually kind of what one of these papers is about. So not only are backing up a little bit to the idea of gene activation, not only are genes like switched on or off, oftentimes their state of being on or off needs to be maintained. So you need to keep producing a molecule that will keep it on. Or in the case of some genes, you need to keep producing a molecule that keeps it off. Right? I'm just going through the bullet point of like things that like I should have said at this point, but I didn't. But a lot of genes, actually, and a lot of molecules, their only function is to kind of signal these processes. their only function is to interact with this process of turning DNA into RNA, known as transcription, and regulate it in some way. And the genes that do this are called transcription factors, right? This is very useful for kind of consolidating functions. Like if you want to turn on a bunch of different genes at once, you can have one transcription factor produced that then essentially has the function of thousands of genes that signals downstream. Or you can produce one molecule that isn't necessarily a protein, but it interacts with a protein and then causes that protein to be a transcription factor that activates thousands of downstream targets. What are some wonderful examples of molecules that interact with proteins that then activate thousands of downstream target genes? Hormones. This is exactly how hormones work. This is kind of exactly how HRT works. This is getting a little off of the papers, but like this is, a huge thing, right? So every single human being alive, with a very small handful of exceptions on the sex chromosomes, but I can talk about it in a moment. Every single human being alive has all of the genes required to make essentially the full male or female, quote unquote, body plans, right? It is just a matter of one signal in very early development that is correlated with the Y chromosome, determining essentially what wakes up. So you might have heard of the gene SRY. That's the signal I'm talking about. It's a gene found on the Y chromosome, which starts testicular development. Once testicular development starts, it starts producing androgens like testosterone, and testosterone then wakes up all the other sex characteristics that we associate with quote unquote male. If that doesn't happen, then you get, if SRY is not active, you get ovarian development, you get estrogen signaling, and estrogen wakes up all of the traits that we associate with female. Now, this usually happens in utero. And for a very long time, it was thought that past certain developmental markers, a lot of that development was completely locked in. That once you reach this fully differentiated state of having, say, I'm going to use like transmasculine HRT and like, I know this word is loaded, but like quote unquote natally female individuals as my example here because it's the focus of one of the papers. But it's, for example, if something is fully differentiated into uterine or ovary tissue, it was thought that was locked in, right? You can't do anything to it. But as you may have like already seen, I've laid the seeds to be like something that is fully differentiated isn't necessarily locked into that state forever. Which brings us to one of the papers, pretty recent, so a lot of the actual mechanisms of it are still poorly understood and still need a lot of follow-up. It's from 2021. This is the prostatic metaplasia of the vagina in transmasculine individuals. Basically, so the TLDR on this whole process, this whole prostate, this whole process.
I can never make my way through a whole prostate. It's it's a bit of a drag.
It's way up in there.
Yeah.
All right, too long didn't milk of this whole prostate.
All right, we milking prostates in here.
Real briefly on this because I do not think I think this is going to be much more dense than a topic than we will have the time to cover on the remainder of this podcast.
I'm literally. Can I give like one sentence summaries of each of the papers that kind of build?
No, I want to hear this. I want to know. I want to know the build up you were building up to.
So the TLDR of the prostatic metaplasia paper is basically that they did a bunch of like hysterectomies on like, transmasculine people who had been on testosterone for a very long time and then wanted hysterectomies as part of their like transition or bottom surgery or whatever. And they looked at them and they found inside the tissue of like the uterus and vaginal lining, they found disorganized prostate tissue developing. In all of the samples they collected.
What?
And so it is, the actual mechanism is unknown, technically. However, based on what I've seen and based on some things I've seen in the field, it is thought that this could be an example of transdifferentiation, where you induce this change in signaling, this change in a signaling molecule that is related to defining a cellular fate. And it actually is able to cause a change that normally should be locked in from the time you're in utero. So it's disorganized prostate tissue. It's not like actual like prostate developing, but that is like really, that I don't think I need to tell you that has really exciting implications.
Yeah, that's huge.
You can actually develop these tissues and maybe encourage them to kind of organize themselves into a particular way. These tissues do perform basic functions like producing even more androgens on their own. So yeah, again, way more research is necessary. And unfortunately, some of the actions of like scientific funding bodies in the past year mean that like this follow-up research is likely not going to happen anytime soon. But this is absolutely fascinating. Now, in the nature of fairness, the reason I provided 2 papers, let's talk about the girlies. So I mentioned that like cellular fates not only need to be determined, but they need to be maintained. And so I don't think it's actually ever been studied, like the effect that like estrogenic HRT potentially has on this. So I won't tell you anything one way or the other. However, there is this gene known as DMRT1, which is constantly produced within the testes. And the function of DMRT1 is to keep the testes. It is saying, testicle, you are a testicle. Testicle, you are a testicle. It is reminding the testicle that it is a testicle throughout its entire life. Those balls stay balls, damn it. But it's been known to have that function for a while. And so, in this other paper, the authors were just like, hey, DMRT1, shut up. Like, stop talking. Stop saying anything. Stop saying the testicles are testicles. And what ended up happening was that the testicles started taking on some ovarian traits. They started
Holy shit.
reverting to a less differentiated state and possibly following and expressing some of the differentiating factors that are associated with ovaries. And they started producing a little bit of their own estrogen. So,
That's so sick.
it is so
It rules.
fucking cool.
That is incredible. Even just beyond the implications, like, for us and our healthcare, that's just A, really cool, and B, has huge implications for, like, possible, like, future narrative developments in professional wrestling.
Oh, yeah. Yeah.
Yes, but
Ray Mysterio pregnant arc?
Ray Mysterio turned his balls into girl balls.
oh my god. Right. 1, for our healthcare, but 2, I think, like, to be real for a second, if that, like, showing what is possible there, like, completely disintegrates this, like, bioessentialism, like,
Yes.
you are born this way, and, like, you can never be X, Y, and Z that, like, TERFs say, and this kind of thing. It's, like, literally, it is possible for any body that is a human to do this. That's wild. That's like huge. I feel like that's huge for like the argument, right?
The possibility of endogenous hormones for
Oh my god.
yeah. Like that's crazy. That's cool. Okay. Yeah. Wow. This has been an exciting episode of Bimbo Mentality. Not gonna
I
fucking
know.
lie.
I don't want to oversell this, right? Because
Right.
like this is like what we're talking about is like the production of some disorganized tissues. Like there's cells still within the old cells that are taking on some traits of stuff. And any functional application of this is, you know, the kind of thing that's like decades out, and especially considering the controversial nature of this research, like I don't want to fucking call it controversial. I don't want to call it. Considering the hated nature of this research,
considering the suppressed nature of this research, and the
Considering
degree to
the-
which people are deliberately going to try to keep us from researching this because people want to hurt us, and they can't stand the idea that their pretense for hurting us is bullshit, because if it is proven scientifically to be bullshit, which it obviously is, then people will just have to sit with the reality that they're monsters who like hurting people.
Mm-hmm.
Yeah. I mean, yeah, like, so this having implications for trans healthcare is probably pretty far off, like direct implications at least. This is like not the only context that like trans differentiation is studied in. The trans is just a Latin prefix that means like, you know, different. So it's about the jumping from one cell type to the other. And I did actually, as part of my grad school research, like study gene regulation in a like non-gonadal trans differentiative process. And so a lot of it is like moving forward with like the core molecular mechanisms of like, okay, what can change? Like, why does this happen sometimes? What situations allow trans differentiation to happen? What's happening on the genetic level? A lot of those studies are moving forward, especially under the justification of like regenerative therapies. Like the way I always like to spin it is like, this is stem cell research without the stem cells, you know? This is the ability to reprogram cells without dealing with essentially deprogramming them, without dealing with reverting them to a stem cell state and doing all that messy stuff. This is telling the cell, hey, you're this now, and it just doing it. Also, as far as like destroying bioessentialist narratives, like I don't want to get like too sappy about this, but like, you know, I was beat into my head so much as a kid that like, oh yeah, you're biologically male, blah, 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 blah. And I transitioned. I like started HRT at the point where I had already been studying biology for 7 years. And those two processes are like not unrelated. I consider studying biology to be a huge part of how I unlearned my bioessentialism. You know, I want to preface this by saying like, this is not a fucking trans med thing. I'm not saying this defines you, but like, when I started learning about like dynamic gene regulation and the fact that hormones do change your gene regulation, and the fact that genetically everyone has like most of the genes required for male or female development, and they're just like switched on or dormant. When I kind of realized those things, and that kind of, yeah, okay, hormone replacement therapy is a biological change. Like, what are you talking about that, like, trans women are biologically male? That was, like, kind of the first lie to me that then caused the, like, cascade of the cis-normativity to, like, collapse in my head. And then that leads you, and once that collapses, not only does it, like, help you accept trans identity, but it leads you to interesting research places, I think, and, like, interesting biological problems that you never would have stumbled across if you kept on thinking like that. And that's, I think, part of what like these two papers represent and what applying biology in this context represents.
That is all very, very cool and very, very interesting. We are kind of pushing up against our we've been going a little bit longer than we usually do, but I do want to quickly...
I'm sorry for being hot and cool and smart and talking a lot.
This was a fascinating conversation. You have nothing to apologize for. But I do want to make sure we stay on format here. So Emma, tell us a little about what you've learned today.
um, deciding which way to go with it. I'm learning that I can treat my autism at a place in Mexico with stem cells. Actually, I've learned that.
Cancun, which is beautiful.
I've learned that I may have jumped the gun on getting rid of my testicles before like I could turn them into ovaries. And
I assume they just like have them in a jar somewhere. Or you can use the stem cells to regrow them, and then you can use the stem cells again to turn them into ovaries.
Okay, I'm looking to get rid of mine, and like I want to keep them. You know, I want to like do stuff with them. Like,
if I gotta turn them into like external ovaries, I might have kept them even with vaginoplasty and like just gone with it. Like,
Fuck
I just had a really bad shot yesterday. It bled everywhere. So, like, this would have been, you know what? If I could make my own, that would have been pretty cool. That's what I'm saying. That's what I learned.
So many, like, cool, fashionable, like, you can, like, use them as kind of an accessory.
Oh my God.
I was thinking, like, you know how people have Newton's cradles on their desks?
Yes.
Yeah.
Like, get a couple, you know, you get 3 people, that's 6 of them.
Oh my.
3 T-girls, just...
I have learned a lot about... I've learned that wrestlers are now immune from ghosts. I've learned that you can use stem cells both to make people more trans and more autistic.
Yes.
I have learned that there are certain kinds of cells that just kind of like hang out in your bone marrow and like kind of like play Call of Duty and don't really like have cell jobs. I did not learn that the entire pretense for transphobia and bioessentialism is complete bullshit because I already knew that. But I did learn a little bit more about how much bullshit it is. And, you know, sometimes the point of the pursuit of knowledge is like as much about just sort of making a kinder world and ideologically running over shitty people, and you know, reality supports kindness, and trying to outsmart your need to be kind and loving to people is bullshit, and that's Bimbo Mentality, baby. Sierra, having heard what we've learned today, do you feel like you have successfully thought like a bimbo and taught us about the field of molecular biology today?
I feel pretty good. I'm very happy, you know, I really love chatting with you all. Yeah, fuck J.K. Rowling. And yeah, thank you so much for paying such great attention and being such nice hosts.
This was so
It's been
fun.
an
I
absolute
learned
delight.
a lot. Yeah.
It's been a delight having you on. This has been a really, really interesting conversation. Do you have anything you want to plug or anything before we go?
I don't know. I'm still on Tumblr in the year of our Lord 2025. So
Fuck
Catboy
yeah.
Biologist.
It is 2026, by the way.
Oh fuck! Right.
Oh, now who's out bimboed by the Bimbo the Bimbo?
Oh my god!
Wow.
Fuck. Okay. Anyways,
well, we give a
Catboy Biologist on Tumblr and Reddit. Hi Sierra on Instagram and Blue Sky.
Cool. That's gonna do it for us this week on Bimbo Mentality. We'll see everybody in a couple weeks. Bye.